if we could somehow distill biology to the most fundamental blocks and atoms and then simulate it, then the idea was, well, then we could answer any question.
Totally. Biology is complicated. Right? You put your finger in, like, anywhere you go, you put your finger and it extends 1,000 miles, and you're like, oh, it turns out these tiny effects and like buffer liquid flows change how the cells grow, etc. Nothing is real.
How can we simulate it? How can we understand other parts of it broadly? Is kind of the vision of our company, and that's what we are building.
Everything we do in drug discovery and development is almost all a simulation or approximation, right? Like doing a mouse study is an approximation of humans. Doing a cell study is an approximation of a living system, right?
If you look at aging, there's not going to be one or two drugs which there's not going to be this magic pill you take. You have to look at different tissues, what's going on, many different processes. So you probably have to have a very multidimensional, multifactorial interaction. We are building a platform. One can think about it as a drug discovery or biological OS. It is has a number of components which help researchers at different stages of their work.
Sounds like science indistinguishable from magic.
Well, that's what we're aiming for, right?
Hi everyone, it's Renegade Science podcast and your host, Marie-Neve here. In this podcast, I meet scientists and entrepreneurs that are creating groundbreaking technologies that will eventually change our world. I talk to them about their work and areas of expertise and ask all the burning and stupid questions, the most important of which is, how do we build a great future powered by tech? Don't forget to subscribe to our YouTube channel and social media. And today, we embark on a journey towards the origins of life. No, literally. Although humans spent centuries trying to figure out how biology works, we still do not understand it fully. My today's guests want to change the status quo. Their goal is to simulate life, starting from the smallest building blocks, atoms and molecules, and by that, gain insights that not only will help modern scientists in drug discovery, but in the future will allow to solve aging. Please welcome founder and CEO of Deep Origin, Michael Antonov, and Head of Business Development of Deep Origin, Natalie Ma. And we are live. Hello, Natalie and Michael. Thank you for being here.
Hello, thank you for inviting us, Maria.
Thank you. Thank you for having us.
My first question would be about longevity. I know that both of you are interested in longevity, and I'm curious to know, how did it start for both of you?
So I guess for me it started about 10 years ago. I was in Facebook at the time. We just exited Oculus, which was our first company. I was trying to figure out what's the most impactful thing I could do. And at that time, I actually ran into Aubrey de Grey, who was presenting about biology. And he kind of inspired me to more independently look into essentially biology of aging. So genomics revolution was happening. I was trying to figure out how deeply can we understand ourselves and our bodies and what actually forms aging. And the question was, can we actually do something about it in our lifetime? So that's really the kind of key area which I got interested in. And ultimately I ended up learning, studying science while still working and investing in the space and really kind of going from there.
Well, yeah, that's impressive. I read that you actually spent several years studying biology.
Well, I haven't studied as seriously as the real PhDs and people who do it. I was working then, and I had an opportunity to go to lots of classes at the Berkeley Extension, and I went to lots of conferences in longevity and kind of got involved in the community, which is really an upcoming and passionate group of people. And I got very inspired. Oh, maybe we can actually do something about it. Then, of course, you realize that it is quite complex, and there's so many also holes and gaps. And as an engineer, I was hoping we could actually simulate life. So imagine, as a programmer, we and like at the very bottom you have this assembly language, which is just a lot of machine instruction, and then you get all these kind of high level systems on top. Well, if we could somehow distill biology to the most fundamental blocks and atoms and then simulate it, then the idea was, well, then we could answer any question. Of course, that dismisses the big gaps in knowledge and the state of quantum effects and so on, which actually makes it a lot more complicated in biology than it is in, real, well, than it is in computing, which is much easier.
Yeah, that's true. Absolutely. And I'm sure that Natalie would add something to that immediately.
Yeah, totally. I have to say, Michael is being very modest. For anyone out there who wants to like induct Michael into a full thesis program at this point, he's pretty much ready to get the PhD in biology or biotech. Yeah, in terms of my interest in longevity, I sort of stumbled upon it. So previously I was working in infectious disease. I know, Maria, you and I have previously chatted about some of my earlier work and crazy adventures in backpacking around the world. But after my last startup, I was thinking about, how do we develop drugs? I'm a person that I suppose you could call a failed drug hunter at this point, right? I took an asset to phase 2 and it didn't work out. And so it's like, if this is the state of biology, right? Like it takes us so many shots on goal to make a drug successfully. If you think about things that truly extend health span, as Michael mentioned, the feasibility of testing any of those is nearly impossible, right? Because it's like you have to run so many trials to get one success, and each of those trials will be a 30-year timeline because you're looking at an extension of human health, and it becomes an untenable problem. And so, when I had met Michael and he was talking about this idea of the simulating life, right, starting Deep Origin, I was, of course, very skeptical initially. Again, as a person who came from the wet lab, right? It's like, this is a place where frequently I'd say people from tech have come in and had to eat very humble pie, right? Because biology is complicated. You put your finger in, like anywhere you go, you put your finger and it extends 1,000 miles, and you're like, oh, it turns out these tiny effects and like buffer liquid flows change how the cells grow, etc. Nothing is real. Wet lab biologist here. But like as we started working together, particularly with like the broader team at Deep Origin, like I realized that like, yes, there is a tremendous amount of nuance here in modeling these things. But I think Michael and Georg Papayan, our CSO, and like the broader team has done an amazing job understanding and respecting those nuances and being like, yes, that's a problem. Let's go allocate resources to understanding why that's happening. Not like, yeah, that's a problem and we'll sort it out later, but like tech will fix everything. And so that was very like super heartening for me, right? Because I think there's a lot of a perception that is the case when tech gets leveraged in bio, like tech will solve everything. I think in this case, we've been, pretty good about thinking about, okay, where are the places tech can be useful in biology, not just applying tech for technology's sake.
Actually, I see that in the last years, longevity is slowly moving from the fringe to the mainstream. And I wonder, why do you think it's happening?
Well, it's been evolving a lot, I would say, over the last 15 years, because in the beginning, I think if you roll back to 20 years ago, if somebody would say longevity, then often, even if you're a researcher, I've not personally experienced it, but I've heard that scientists would lose credibility if they kind of go down too much down that path of thinking. And it has gradually gotten more investment. There's been a number of funds which specifically focused on longevity. There's an active community. And increasingly, there's been more and more investment which goes into the space. And also there was the development of biomarkers, which is all these epigenetic clocks and different ways to track the body, which have given people some measurable reference, which we can try to optimize. And overall, it altogether accumulated to much more active community and acceptance. Also, even with that being the case, I would say many longevity companies still end up being therapeutic companies. So they will often pick maybe a research path in the beginning, which relates to some kind of age-related general cause. But for FDA to approve it, they need to pick an indication. And beyond that path, it really becomes a more classical therapeutic development. But the idea here is if you develop therapies that are, based on the mechanisms which more generally can prolong lifespan and health span beyond that one indication, then maybe it can be more broadly useful. And then, of course, there's a whole testing, for an optimizing health and supplemented the extra industries which exist. And some of it, I think, will gradually help more and more, even though a lot of it one can be skeptical about individually.
I think, Michael, you hit the nail on the head. I don't really have much to add except for, in this place, this is where mad props, I think, goes out to academia, who started taking aging more seriously, right, as a paradigm, and understanding how all these diseases that we consider disparate, right, diabetes, fibrosis, autoimmune diseases, arthritis, thinking about a unified paradigm to add them. This is a place where, longevity, I think, Michael, correct me if you think differently, but I feel like it gained legitimacy because academia put out a set of papers saying, hey, there's actually a substantial number of connections between a lot of these diseases, and there might be some sort of set of things that drive aging, right? There is differential aging between people. There is differential disease profiles between people, right? And so the academic world did some amazing work there.
Yes. I mean, there's definitely more and more connections, and people see how aging really leads to huge number of diseases. Also, I think there's a couple other factors. We got a few closer to clinic progressing companies. So, for example, Biotech, APOs, and that really focused on longevity. So that's really just recently, I think last week. And I know in Silicon Medicine has some good progress in their assets. So there's a number of companies who kind of came around that time and supported by the longevity movement and have been instrumental to that. So I think that combination of some people making it to later stages and bigger community and more general biological understanding of how our state changes with age is what leads to this greater optimism.
That sounds exciting. And still we need to simulate life to arrive to the point when we can finally understand what happens, to the body when we are getting older. So tell me what journey led to creation of Deep Origin and actually why you decided to take this name.
Well, I think we have already touched on simulating, which was kind of a basic concept. And I've sort of come to a conclusion that thinking about systems and science, at least for me, is if we can't really model something, we can't understand it. Which really is, if you just have a cell and you run some experiments and something changes, you put this, whatever chemical in and some behavior is different. I mean, that's maybe useful outcome, but if you don't understand the steps, then you don't know what else is going to affect it and how does that work in a bigger system. So I think while we probably won't be human simulating life with cognition and all this advanced levels, we can get pretty far, I believe, in cells and tissues and in the future organ systems to a large extent. So my kind of real interest has started sort of closer to the cellular level, just because we don't actually understand, or we can't model very well a lot of the biochemistry which is going on. So, and biochemistry relates a lot to drug development. So, for example, if you have in the classical system, you have something called the target, which is maybe a protein, which is implicated in some disease. And now if you could somehow block that protein from, doing what it does, then we can do it with small molecules or other ways, then maybe the condition would be changed and improved. So that's the essence of it. But how do these all things fit together? How do different proteins form together to do some kind of more general function is something we've done researched a lot experimentally. So we do lots of whole range of experiments from like crystallography, which freezes, which organizes things to try to figure out their structure, from like cryo-EM to more basic biology and observational state. But we don't get to the bottom details. We don't see the dynamics of the molecules. Therefore, we kind of have to guess, and then we have to run computational methods. And the idea of the question was, how do we get to the kind of broader company and simulation of life, is that could you actually build these models on multiple levels? So could we simulate things out of atoms to predict molecular structure, and then take that next to the level? Okay, now these kind of complexes can form broader, maybe membranes or different parts of a cell. And can you combine it with also enzyme kinetics and the way genes respond? So you can build this multi-tiered system which has feedbacks into each other. So if you could do that in theory, that maybe we can simulate the cell, maybe like a human immune cell, an example, and it would respond to different stimuli and to different external conditions and the molecules in a way which is similar to a real cell. So that would be sort of the meta incredible project, which I think if you did that accurately and you let people measure parts of this, you could get to deep understanding biology, but also support a lot of therapeutic uses. So really thinking about that vision, what would enable that, which is still today not something we have done as a humanity. The idea was going to these basic, deep binding blocks, such as molecules of life, and which kind of and everything assembles and originates out of it, is sort of my thinking out of the name Deep Origin, is kind of going down to how biology works internally. How can we simulate it? How can we understand other parts of it broadly? Is kind of the vision of our company, and that's what we are building. So, and also a lot of computational tools which help you do the discovery, understand it.
Well, Natalie, how possible is it to actually simulate life? Because it sounds amazing. Yes, this is a quest for the origin of life, for how life actually works. Maybe you will even find a reason, an answer to everything, and it won't be 42.
It might be.
Yeah, maybe we still don't know. We haven't built it yet. You won't like this. But Michael, when you were talking about sort of what were you describing, if you can't model, we don't understand it. I feel like we need a picture of Feynman up somewhere in the office. There's this great, probably apocryphal story of him going to the library, I think when he was at Oxford, and asking for a diagram of a cat, right? And realizing that there's no way, we don't have any models of life. And so this is where what I cannot create, I do not understand quote comes from. But yeah, I feel like this is very reminds me of very Feynman-esque things. I think is it possible question, putting my wet lab biologist hat on, when I got my PhD decade ago now, oh boy, 6 to 8 years ago, somewhere in there, it was probably at the edge of viability in the sense that data collection was picking up. So there's a couple of things you need here. And we have, to give a shout out to the folks who have done this already for decades, we have been trying to model different aspects of life and succeeding in many places, these sort of point parts in the landscape, as Michael mentioned, for 50 years, right? I think 1961 or 1967 was this big, cover of some famous magazine where it's Merck was modeling some protein computationally to try and find a drug to hit it, right? The main thing is that people have been focused on these specific things, right? How do we get a binder to stick into a protein? How do we understand the folding of a protein? How do we understand how the gene expression changes? So going back to what I started with, you need a couple of key things here. One of which is the models themselves, right? And there's a couple of different flavors of models. There's a physics-based, perspective or first principles or mechanism where it's you define the rules of the system. And you have sort of machine learning models where it's like, we don't fully understand like the rules of the first principles of the system. We can throw a bunch of data at the problem and get a bunch of like correlations, right? Trends, understand, or like, okay, if A happens, then B happens, right? And so besides the models, the other thing you need is data, right? You need data to validate the physics models, right? Understand them. You need data to both train and validate the machine learning models, like tremendous amounts of data. And that's where, 8 years ago when I graduated, we're starting to get to that point where it's like, you want to sequence something, get a whole genome sequence. It wasn't as much of a problem. When I started grad school, that was a cost prohibitive thing you wrote an entire grant to do. Then it was, by the end of my grad school, 5.5 years, it was like, oh yeah, throw a couple $1,000 to get a whole genome sequence, really short content, throw it on the sequencer. It was a much, like that cost came down exponentially. It was really impressive. And so we have like this volume of data we can collect. The problem is, as I mentioned in the last time we chatted, Maria, was like people were not collecting this data for a specific purpose and not attaching the metadata to the data. So you get piles of data on the internet, and it is not designed in any way for you to do machine learning on it. I know there's lots of people in tech who are in tech for biotech who are enthusiastic about building models. My hot take is we don't have enough data yet. We are not close to that, but we now have the steps or the tools in place to do so should we choose to do it, responsibly and with all the concomitant information required to build these models. I think we're also of the philosophy that there's not going to be a single unified model of biology, or at least I'm of this opinion. This is hundreds of interconnecting models, right? Each of which is designed to solve a specific problem, whether it's physics-based model or mechanistic model or a machine learning model. But these sort of collection or ensemble of these models is going to be what gets us to this place of like atom-atom interaction here between small molecule and a protein, to a change in, protein dynamic gene expression, all the way to a change in cell physiology. But like the individual pieces have been around already, and mostly it's, making them more accurate and stitching them together, which is what we started doing recently. We actually, Garrick, our CSO, just showed data at a Discovery on Target last week, indicating that, our all atom and coarse grain models, these small molecule to protein and then sort of protein dynamics could be put together and outperform AlphaFold 2 on prediction of PROTAC structures, which are these protein small molecule protein ensembles. So that was really exciting. So we're starting to do that work, and our future goal is, okay, can we stitch more and more of these models together? We're at the same time sort of driving revenue and business with each of the individual tools.
Yeah.
Because we have to be a for-profit company.
So yeah, I think the kind of short way to summarize some of it is that there are particular levels of models where we can get meaningfully accurate results for short durations or limited durations of time. And so, for example, some protein interaction you can do with molecular dynamics, but it has limitations. Some pathway analysis you could do with, kind of called kinetic equations, and it has its purpose, but then it has its limitations. So the big challenge is how can we grow these and make them broader, like Natalie described, where we have PROTACs or some kind of a bigger system which is accurately represented, and we take that and repeat and integrate that. So the answer is some things we know, but there's lots of connecting which needs to be done.
It's true. Yes.
And I would say AI is sort of this wild card which comes in on top. And on one hand, you could train database models, which could be very predictive. So it could actually, one can imagine if you have enough data on a higher level, you could predict a lot of things without even understanding what's going on underneath. At the same time, there's going to be conditions under which that doesn't work. And it's also even hard to collect that much data in biology, so industry is certainly focusing on that a lot.
Yeah, now I want to ask 10 questions.
Go
in
for
the
it.
same time. Yeah. So first question is that like general big LLMs, yeah, they are super expensive to train. We will soon run out of available data to do that. In biology, this is also an issue. And you said that there is not enough data, but what are there any other specific challenges that biology is facing in terms of using AI?
Michael, do you want to take this or should I?
Well,
I'll pitch cost per data point. right? Like
Yeah, yeah.
it's free. It's not entirely free because you have to factor in the cost of the Twitter engineer. Although Elon Musk fired most of them, so maybe it's much cheaper now. But it's basically like the cost of getting a sentence with some context, right, on the internet as a data point is fairly cheap, right? We generate lots and lots of those, and people are doing it as part of, social media. Sorry to the Twitter engineers. The cost per data point in life sciences is not that, right? You have to expend effort, and not just effort, but tremendous effort of scientists, and then the assay reagent costs, and then the actual assay run, and then the analysis to get any data point out in biology. It's like a fundamentally different scale of data cost, right? So you have, you have a world in which you can I definitely bring down data costs, and that's part of our goal on the infrastructure side of our company, right? It's like, how do you build a system that is much more performant in the hands of a wet lab biologists and bioinformaticians, and then collects the data as seamlessly as possible with the concomitant metadata, right? The labeling, anything that you would need for future use, both as a reference for your company, right? I need to look something up, as well as building a more comprehensive model. But that I think does have a floor. It will always be cheaper to run a simulation than it will be to run a wet lab experiment than it will be to run an experiment in people. I think that cost scale, there's not much that changes that cost scale at a fundamental perspective.
Yeah. I think we need Amazon scale large robotic labs with some kind of, I don't know, organ on chip microfluidics or whatever it is, so you can also and tie it all into computational analysis. I think that would be kind of a good platform. I think before we have some really major, huge projects like that, another challenge is consistency and quality of data, because each individual data point is hard. That means different groups and organizations do it. It's often done inconsistently, which means it's kind of harder to actually interpret and get as quality results out of it. And also there's very many-dimensional space. So it's much more challenging in every single aspect, I would say. In terms of whether we have enough data or not, I've actually heard different opinions. Occasionally I meet people I respect who say, well, there's actually enough public data. If you do this, you'd be surprised what you can do. So we probably haven't extracted everything out of what's available. That said, I would say we are missing a lot of core understanding at biochemical level, at many other pathway levels. So, for example, lots of progress with AlphaFold was based on existing databases, which they could trade in. And then there's aspects of that, let's pick disordered proteins or other structures, which we don't necessarily have as much understanding and databases for. So therefore, things in between, those kind of models are not as strong as predicting. So if you really want to do that, you need another approach, maybe physics or whatnot. So I think the reality is that in biology, we definitely are short on data, especially for particular use cases which you want to understand well. And that's when, if you're really going to scale a focused project in a given area, every organization often says, okay, at this point, beyond our kind of doing the introductory work, we need to build our own lab infrastructure to do consistent experimentation in this direction to truly collect the resources we need. Because then people can be more confident about the quality of it and hit the necessary scale.
Yeah, I mean, look at like Recursion, right? Recursion spent all this capital to do the RxRx data set and their internal data set. DexDNT, I think, has done something. Now Recursion has done something similar as well. And they've both done almost fully automated labs, I think, Michael, as you said, for consistency.
Yes.
Sarah, go ahead, Maria.
Yeah, I already thought maybe somebody will spin off an Amazon for producing biodata. I mean, at least last week I interviewed guys who have a project. They're connecting biobanks all around the world. They're called amino chain So, and they're connecting in a decentralized network biobanks all around the world and building actually an Amazon for biobanks. So who knows, maybe we will have an Amazon for biodata at some point.
That would be amazing. I think we have some initial plans to like host data sets of utility. And there might be some direction we go in the future that also relates to that, right? Because it's like that I think has a tremendous amount of value and is actually very hard. Like access to data sets, even the publicly available ones, can sometimes be super challenging. It's great that they're working on that for biobanks because that's all finding the right tissue sample or the sample is a huge problem in this space. There have been a couple of players that have tried to do things like in a similar angle, like for like research, like mostly CRO focused, like aggregation, like services. But I don't think I've heard anything biobank related, so I hope they're super successful.
I think they're quite young, but we will see anyway. And I have a stupid question. Well, actually, my question was stupid before because I wanted to ask, can you use synthetic data to train biotech, biotech models? But now I want to ask if you can simulate with like high certainty some biological processes, can you create synthetic data and train biomodels on it or not?
Up to a point. The answer is in some cases yes, and it has limitations. So it's I think the challenge is, so for example, you get fairly developed field of molecular dynamics where people have doing some kind of atomic simulations for a few decades, and they've built these energy fields for different molecules, and these fields are used to evaluate what are the conformations which are possible and how things might evolve. And there's going to be inaccuracies in those fields. And when you have novel molecular structures, some of those fields may be even less accurate. And the reason this happens is because there is a much more complex quantum chemistry, which is very expensive to simulate. So we can't really do compute at reasonable biological scale there. So with that being the case, what we can now try to do is build learning by running the higher quality model for some period of time and collecting the data, maybe you're training neural networks, and then you're using that at the high level to get quicker readouts, therefore you can do broader range of experiments. That said, there's going to be cases where you will get either accumulated error or unknown because of the imprecision in the system. So it's sort of one of these things which can help you take one or two big steps, but there's going to be a ceiling. And once you have a ceiling, you need experimental data to correct it, and then you have to figure out how this whole stack fits together.
Yeah, I mean, I don't think I have anything else to add except for like the curse of the experiment. So as the person who is the wet lab biologist, like you have to realize that everything we do in drug discovery and development is almost all a simulation or approximation, right? Like doing a mouse study is an approximation of humans. Doing a cell study is an approximation of a living system, right? And so you do get this like compounding error question that Michael, I think, alluded to, right? You have a limit to the accuracy of your system. And when you hit that limit, like if your training data has some intrinsic error in it, that error is learned by your model. So even in like, yes, with simulated data, you have like this concern about like the limit of sort of not knowledge, but sort of like the accuracy. But I think that also applies to experimental systems, right? Like there is going to be noise in your experimental system as well. The question is, I guess, like how much noise, and to your question, Maria, high quality / highly accurate is the underlined word in that sentence, right? Like if you've got that, then yes. And if not, then no. But you very rarely know whether that statement is true, like whether that statement is true, right? Is your data set highly accurate? What is the noise in your data set? I think, sometimes, not always.
Looks like life is really complex. And now I want to go back to the drug discovery because your company, Deep Origin, helps to speed up the process of drug development with AI. But first, I would love to ask for drug discovery 101. Yeah, for people who are not a biologist. So I want to create a new drug. So what I need to do?
Oh boy.
Eventually.
Yeah, I'll take this one. As the like I said, the failed drug hunter. So the process is probably we're talking 15 to 20 years first off, right? And you have two big groups, right? There's the before the clinic we call preclinical, and then there is in the clinic, which is clinical, and then at the end you have this milestone, which is like an FDA approval, which is in the US the Food and Drug Administration, in the EU it's the EMA, and then there are some other things that also happen because each of the EU member states has something slightly different. But they say you are allowed to sell your drug. We believe the claims that you have made about it and its ability to treat a disease. You are now permitted to sell it. Like that is what you're aiming for as a therapeutics company, is that approval in some way. And so everything is leading up to demonstrating that. And to demonstrate that, you run probably 10 to 15 years of clinical trials, a phase 1, phase 2, phase 3. They come in slightly different flavors. You can sometimes combine them, like a phase 1, 2. But the things you're trying to test are in the beginning. Is it safe, right? Is this safe to give to people at all the doses you would expect them to take? Or do you see any adverse effects? And if so, how do you manage those? And then the phase 2 and phase 3, is it effective? Does it do what you think it does in the disease? And like there is a lot of value in there being a governing agency, like a third-party government agency like the FDA or the EMA, telling you that this is safe and effective because you have a neutral referee that then gives you validity in the system, right? Before we had these, you can look at the world, it's a very different landscape. So everything in the preclinical is leading up to how do we maximize the chance of clinical success, right? Because the success rate out of phase 1 for your average drug depends on the indication, but it's about 15%. So a little over 1 in 10 drugs will make it through that gauntlet to success. And that is an abysmal failure rate, given that the cost of that stage is hundreds of millions of dollars. And if you see any signal anywhere in there, oh, it's not safe, oh, it's not actually effective, okay, it's back to the drawing board. You've lost your investment of between 500 to 700 million. So preclinical is like 3 to 5 years, typically, can be a bit longer, shorter. It itself can be $100 to like $500 million in cost. And again, your goal is to set yourself up successfully for clinical. It's divided into sort of like 5 or 6 major stages. The first of them is you have to find a target to drug. Do you have a thing that you think you can hit that will change the progression of disease? So you need to understand the biology. Second step is, okay, you've got this target you think you can intervene on. Where on that target should you be hitting? What do you understand about the target biology? That's like, yeah, we need to change it to this shape, or we need to stick something in there so it no longer works, right? Like just sort of jam it and like sort of put a wrench in the gears. Which is how most drugs work. They're almost all inhibitors. And so you then have that sort of target characterization. The third step is you then look for something that meets those criteria, like a small molecule, an antibody, something that will do that, have that effect, that sort of hit finding, hit ID, hit design. And then you embark on this process of, well, gee, actually just affinity does not a drug make. You have a ton of other properties because you're giving it to a person who is a complex system. This is all the things around solubility, absorption, metabolism, excretion, distribution. Like there's a huge number of things that go into that. So you're trying to predict all these different properties and optimize for like sort of like the least worst scenario. Like this molecule has this property, this one's not very good, but I think we can live with that later by changing something downstream. So this is hit to lead, right? So you're trying to find something that binds better and then has some desired properties. And then you go to lead and lead optimization, which is like, again, you're tweaking these properties, like all this toward, can you get a drug that meets all the criteria you want to, or a molecule to meet all the criteria you want for it to be a drug? And then you go into all your preclinical animal models, and particularly IND-enabling tox, because you don't want to give something to a person that might, kill the first batch of people that you give it to. So we test first in usually a small animal model like a mouse or a rat, and then a large animal model like a dog, a pig, the monkey, and so forth. And so that's sort of like the pathway working backwards that gets you to an approved drug. And like I said, there are many people I think in this space who you work in this space your whole life and you still don't have an approved drug because of that failure rate and how long it takes, which is really tough, right? It's like you dedicate your life to this, and at the end, the odds that you have something to show for it are not guaranteed.
Yeah. In the end of the day, yes, you have these kind of two, I think, big parts to it. You have the preclinical, which is all this biochemistry to understanding what's going into animal work to prepare for submission. Then there is a clinical when you're testing in humans, and the clinical is more expensive part. And one could argue that, the clinical is a larger bottleneck just because so many years and the tough process it takes, we don't get enough drugs there. I think improving that is largely a political question. And there is certainly, I think with some organ on chip and such systems, there is some movement and interest that maybe in the future we'll be able to accept more data there. So there's ways to maybe that can be sped up, but it's very much you have to focus on a different side of an industry. And the preclinical part is where we work. And the reason I've kind of wanted to focus there specifically is because holistic understanding of biology is going to come from that part. And if you are, let's say you solved the clinical part, we'd have drastic progress, of course, because the iteration would shrink and you'd be able to get more drugs to market. That said, we really still wouldn't know how is a complex number of perturbation affect the system. So if you look at aging, there's not going to be one or two drugs, which there's not going to be this magic pill you take. You have to look at different tissues, what's going on, many different processes. So you probably have to have a very multidimensional, multifactorial interaction. And so to be able to do that, you really have to have really good modeling and understanding. So that is my take is needed for us to truly solve aging. And that's the type of technology I wanted us to focus on building. So that's a reason we are in tools in this space.
For me, there's a description of first you want to understand the mechanism in which you want to intervene. Then you trying to understand with what you can do that, and then you iterate. It reminds me a lot software development, actually. Yeah, and the development, so software products with iterations. So I definitely see some synergy here. Am I mistaken or not?
No, I mean, I think there are some parallels. It moves a little slower, just a little. Turns out writing the code and getting it out a little faster.
That's right.
But yeah, I mean,
a day iteration versus a year,
Yeah, just a difference. But yeah, I think that there are definitely parallels, Maria. And I think there's, this is aside from like deep origin, there's probably tremendous value in the two industries learning from each other, particularly as they grow closer together and you see less and less of a barrier between tech and bio. So yeah, that's an interesting future thing to explore if anyone's looking for a social sciences thesis project or interested in starting a new company. That's probably, there's an opportunity to capture a lot of value there.
I want to ask Michael, because you are a software developer. Definitely your experience here is most relevant.
Well, it's funny enough, 4 years ago, I wrote a blog about this, how the development process is a cycle, and how you can change and observe a state of a system and iterate on it. I think the real challenge is in iteration rate and what can you observe. So that's actually one of the reasons we want to have simulation tools, because if you're doing it in a wet lab, it's just by default going to be most of the time taking a lot longer. I can think of probably some exceptions at like very narrow class of experiments, but also computational, it's a lot more scalable and it's a lot more reproducible because you're going to get similar results every single time. So all those dimensions, to the extent that we build good models of some parts of the system, those could be drastically sped up their compute. So that's a big benefit here.
So you are going into this process, and helping to iterate faster. You are automating this process of drug development, drug discovery. How exactly you do that?
We're building a platform. One can think about it as a drug discovery or biological OS. It is has a number of components which help researchers at different stages of their work. So we have some tools in terms of the data hub infrastructure for analysis and lab notebooks and the ways to run workflows which help you computationally, which can help experimental scientists and bioinformaticists to get the data in. So that's one part. But then once you get the data, you say, okay, this is maybe a target I want. You need to figure out, well, what kind of molecule might be able to target it. So there is a whole set of tools around the virtual screening category. So you can be trying to dock molecules, and you do that, a few billion times. And you try what's called virtual screening. Like there's existing databases of molecular structures, such as NME, E molecules, where you can take these, actually, I think it's up to 50 billion of different things and try to see, do they fit within the pocket of this drug. Of course, you do that with AI, you don't brute force at all. And you try to see those candidates. So we can speed up that step from I have my target to what are the good likely candidates through our novel algorithms, which we have observed performing better than what we know out in the open, that allow us to essentially do that within, let's say, a week or two, where now you can go do experimentation. And because of this technology, now you might be able to run a couple hundred of experiments instead of a couple thousand. So you're both saving the time and the cost there. And after that, you can, if you have picked molecules you need to optimize, you may want to modify, generate different variations. You might be able to do prediction of chemical properties. So okay, this molecule is not going to be the one you want. So instead of running those experiments, the AI can really make suggestions and help you filter out those problems. So ultimately, we are building these steps to help both with target discovery, the lab process around it, the screening, and then optimization of the molecule and understanding of a system. And there is another part of it is, we talked about simulations. Like sometimes when you pick a target, you might not know how it behaves in terms of dynamics. They would have some kind of opening. Or sometimes you may want to not just think about small molecules, but like, is there protein-protein binding? Is there, maybe you're trying to ligate the two things together, or maybe target something to be degraded because you don't want a lot of this molecule. So there is this whole structural interactions with it. We also have a suite to simulate those. So we can help the company who is trying to do that actually understand what's going on in terms of dynamics and make it much more likely that they're actually able to find a drug and quicker. Because if they don't understand the dynamics, they just have to do lots of experimentation to try it. And often they don't even have a choice of how to proceed. So basically the stack of technologies together, both lets you do some parts a lot more fast and cost effectively, like screening, finding molecules, finding openings, binding things, and support the underlying experimental process of, as you work in the lab, where does all that data get tracked and how do you analyze it? So we're building this holistic OS to kind of speed up these processes and hopefully shrink the time, but also make it much more pleasant and enjoyable to work in the field.
That would be nice. That's always nice. From a business perspective, we're pretty open on how we work with folks. So we do a combination of software as a service for some of our tools, as particularly things that are already out there, or they're alternatives, but they're not great in terms of performance. And then we also do partnerships, particularly with some of our more bespoke or newer tools. And we've been successful in getting both SaaS customers as well as partnerships with up to 40 million in milestones and downstream upside. So we're excited right now. There's not a ton we can announce publicly with who we've been able to work with, Maria. But we've got a few customers, and that's been exciting to see initial traction. And then, we talked about this, Michael talked about this OS and this platform. Like, one of the big challenges is like this access problem where, we spend so many decades training people, right, to get to the point of being productive in life sciences. And we've got sort of an exciting tool coming out in the next, probably by the end of the year, which is aimed to sort of reduce some of those barriers to entry for particularly for medicinal chemists, and access to certain computational chemistry tools. So we can't give you an exact date yet, but that's been exciting. It's already in closed beta and has a couple hundred users. And we've been getting really good feedback. So we're hoping that's also an opportunity to sort of help the broader, not just like, provide cutting edge tools that outperform other existing tools and are more predictive of biology, but also increase the access of those tools and democratize, the ability to do drug discovery and science to a broader group of people.
Yeah, that preview version of the tool is really an AI assistant, which really ties into some of our technologies. So it combines this LLM goodness of how do you easily request something with novel algorithms that we have. So that is which means with like a single request, a few lines of text, you can, for your target, find its pockets and start plugging things in. So it's, there's certainly lots of assistants everywhere these days that do all kinds of things. In our case, the key benefit is that you really get this really strong foundation of the quality of the back-end underlying technologies.
And
not just
more predictive models.
Not just
small spoilers. Small spoilers.
Yes.
That sounds super exciting. First with BIOS, literally. OS.
The BIOS.
Yeah, the BIOS. And with Copilot, that sounds like science indistinguishable from magic.
Well, that's what we're aiming for, right? That's the hope, at least. Well, hopefully not. Hopefully we can explain it better than magic. But when we can get to the point where we are seamlessly thinking about this, Michael, you talk about this a lot, right? It's like, you don't think about the underlying assembly when you code, right? You could abstract a lot away, and like that means you can move faster when you write in Python or any of these other tools or other languages. We are not at that point in biology. We are like not near that point. And in some places, like I feel like we have actively fought that as biologists. Yes, it's important to know how these things work and have a background in them, but to think about those things every single day as a consideration when you're doing your work is a cognitive load. Like there's a cost there, right, that we don't talk about.
There's a lot of awareness one need to have. Like, for example, we have scientists who are very good at looking and understanding protein and spotting how do you position the molecule. And when they interact with a customer, they can actually look and suggest all these variations and like lots of details before you even set up a simulation, right? And an interesting part of it is, on the other hand, we have AI researchers who will set up this agent. They're like, okay, well, this thing can do all these things for you. It can decide like which molecule to put in, and it can run the screening, and it can do the drug. And I'm like, and why do you want people to like provide feedback? And I think the reality, I kind of come back, it was like real researchers want to know the reasons why. So you really want the machine to be helping you, and then you're reviewing each meaningful step. And over time, maybe as some of these things get better and better, people's confidence in the system becomes bigger, and the granularity of what the machine does for you versus what you review and do increases. But reality is you have this collaboration.
Yeah, augmentation of a human with help of AI. But would it be possible somewhere in the future maybe to automate the entire process? Like I need I don't know, cancer treatment, and AI comes and tells me, okay, you need this, and that, and we can experiment. I'm already running it on the background, 2 billion copies.
I mean, that's the dream. And how many drinks / semi-illicit substances have we had before we had this discussion? No, this is so Garrick Petrosian, our head of AI here at Deep Origin, like is of the mindset, I think, that you take humans out of the loop more and more. And like you can do that and eventually this runs more or less autonomously, right? Like, and Michael, you've talked about this too, this sort of like this world in which you can get a truly personalized medicine, like an N of one, which we've just barely started doing with RNA therapies. So you have things like Nielsen, which was an RNA therapeutic designed for a single girl, right, who had a specific exon like deletion issue in her DNA that led to this really horrific disease. She passed away recently, but they've done more of these and created there's a couple of different foundations out there that now focus on these N of one therapies. There is I think a tremendous, like, to our point earlier, social, political, like, there's a lot of other things to solve. From the technological standpoint, there's a ton of things to solve. I, as the experimental biologist, always feel a little uncomfortable with no human being in the loop, right? Like, but is that just me being like, I'm afraid of being replaced? I do think that our product manager, Max Ratnikov here, has put it really well. Like, think of this right now, today, as an enthusiastic junior colleague. They really want to please. They really want to find you an answer. They will get you an answer. It will sometimes be wrong. So like, it does require your best judgment. But over time, like, we should be able to abstract more away from this. And there will be, I think, a period in which, we are able to move faster as a result of this. I don't think there's ever a world in which we stop becoming some degree of specialists. That's just my hot take. I don't know, Garrick, if we disagree, or Michael, if you would disagree as well. Like, I think that, like, you always have to have that expert knowledge, whether you build that by interacting with this AI assistant, you spend decades in school, you spend time in lab, that's still there because you need to know what questions to ask.
Yeah. I think the level of how well it works, it really depends on the specialization of your system. So if, in reality, if you think about it today, if you had some particular class of development, we had the whole RNA vaccines, which has happened with COVID, and maybe that part, you have a reproducible aspect where you could replace, part of RNA and package it and test it. And that cycle gets repetitive. And the more that cycle is understood and developed, the more you can automate it. And at some point, you can bring in something normal. As long as it fits within that pattern, the system does it for you, right? The more novel and diverse it is, the way you have to do research, you have to do the whole different class of experimentation, the more kind of validation and confidence you have to have in the system. So, and this is where, like, maybe in a world of super intelligent AI, it will like do and answer lots of your questions. But I feel that for quite a while, we humans will want to have aspects of control and decision making on top of it. So that's kind of where we are.
Yeah. And I know that we're running near the end of time, Maria. Definitely happy to take additional questions. But I did see, when you sent through, like, the preview list, there was a question about the folks at Blueprint. Is it Blueprint you chatted with?
Yeah.
And, like, why software does not get created in life sciences, which is a hot topic with lots of
may I then read the question? Yeah, the question. So I talked 2 days ago to a biologist who is also a software engineer. Looks like it happens. And now he works for Brian Johnson's company called Blueprint. So, and he shared with me an interesting observation that currently investors in the field of science are eager to invest in building, like, biotech innovations, but not in the development of tools. So what is your take on it?
That is a challenging one. Like, that's the one I've actually presented at Foresight and thought about this quite a bit, because one premise I tend to live by personally is that I believe tools really speed up progress. And you can see that, like, in industrial age, where you had these machines, where you can manufacture and advanced manufacture, and it helped, make drastic jumps. And I think it happened also, like, in the more modern world in South Korea, where it was able to make dramatic progress, and it had to do with these kind of quality machine tools, and actually the fact that they manufacture their own machine tools. So I think that clearly is the case for the rest of the world. I mean, I think a case in point where we actually are investing right now is the AI, like whether it's like the hype of it or it seems to be quite justified, but it's clearly the world has recognized, okay, this tool is so valuable, we should do it. But all of the other tools, simulating physics, combining parts of pipelines, all of those things biologically can really drastically amplify that speed at which people progress. So therefore, if we have good tools, we can have quicker progress. But the problem is that for any one startup, it is huge value is often accrued to a biological asset. So if you think about it, is and as a kind of investor shoes, I would have also startups present to me, and they're like, "We're building the platform, right?" But then once they have the platform, they have like 1 or 2 or 3 candidates. Maybe they'll have a partnership, so with somebody else doing a force. But then this whole once they have these assets, now they have to raise hundreds of millions of dollars to do the clinic, right? And this technological part is actually not so relevant because once 90% of your cash goes into clinical, and the CEO or a board look at the value of a company, that software or tool part doesn't matter, which means we don't share tools, which means we don't build them as much, which means the ecosystem doesn't actually get enough capital. So that is part of the challenge. And quite honestly, the reason, like, it's fairly challenging for tools to make a story about how they'll scale in terms of their revenue and how to really have that impact and to basically have the investor returns. So that, I think, is a fundamental challenge. Maybe it could be addressed on some kind of government or some, like, incentive type situation. But besides that, I think I would love to have also an ecosystem of maybe incentives in us, in the software and tool ecosystem, which would help tool makers capture more of the upside. And maybe that would actually help creating and speed everything up. So that's, as we think about actually our platform, we're also thinking about how do you move the ecosystem forward. So it's a real challenge.
I think if I can just highlight 3 things that are like, Michael, you hit the nail on the head, of course, as usual. Like the 3 things I would highlight is the person who does business development, right? I think about 3 problems. There's a technical challenge here, there's a numbers game challenge here, and then there is a sales push challenge here. The first one is this technical challenge, which is that a tool, in order for people to want to adopt it, has to change the trajectory of drug development, right? You need to appreciably create some kind of value, and often that value in the longer term is improve the success rate on a drug. And your timeline to demonstrating that is a 30 to 50 year trajectory, right? Like, people now accept that computational chemistry methods, computer-aided drug design, like electrodynamics, docking, virtual screening, have value in finding potential drugs. It took many decades to get there, right? So it's like, challenge 1, how do you find a point at which you can demonstrate value that isn't just we got more drugs approved, because that one is almost impossible, right? 2, a numbers game challenge, which Michael sort of mentioned, is you have this pressure between making your own assets and then making the tools available. And the asset has a high risk, high reward gamble to it, right? You put in a bunch of capital, but you could get a huge upside. The tool is modest upside, but high risk in the sense that there is not, most SaaS platforms or most SaaS in the world depends on a huge number of eyeballs, right? You charge for Audible or Spotify $11.99 to 100 million people, and you get the revenue that you need to justify the software team, right? Bio is smaller, right? And the result is that you have to charge a higher price, which people then get frustrated by, and then fewer people adopt, and you end up with this sort of anti-treadmill, right? Schrodinger is at least $50,000 a year. And again, they have an amazing product, but it's who does that price out? And who then uses the software? And there are a lot of challenges there around that. So getting the pricing model right. And the third is like most of the exits or like places you would do business, like deal, like acquisition, like are geared toward buying assets, right? You talk to a BD team in pharma and biotech, and they're like, I understand how to buy this thing. I know how to value it and how to decide, do we want to go or not on this asset? When you approach them with software, this is a newer thing that they're like, go through IT procurement. And again, it's not that they are not good at their job. It's merely that this is what the larger system has decided. Here's what we know how to value and incentivize. The incentives in the system are to drive this. Acquiring software is a cost to us that goes through IT procurement. We don't necessarily think about it from the BD perspective, although there are BDs who do focus more on like platforms and software now. But I think this is a space where like we're seeing a shift, and like the winners will be people who can learn how to adopt not just assets but tech, like enabling platforms that accelerate their companies and make good purchases there. And like they will hopefully drive also a broader change in the ecosystem of how do we value things and what do we value. But that is very much something I think outside of our hands, and we're just going to have to watch in the ecosystem as Deep Origin and keep doing the best with our tools that we can.
Perfect. Thank you so much. We're already running over time. Thanks a lot.
It's just
I would
delightful
be
to
happy
talk to you, Maria.
to continue this conversation. Yeah.
I know we can get drinks. Where are you? You're in Berlin, yeah?
Yeah, I'm in Berlin. So
Yeah.
if you will be
Okay,
in
very nice.
Germany, yeah, just tell me.
Okay, perfect.
Very
We're
nice.
passing through. If you come to SF, let us know. This has been phenomenal to chat with you. Thank you so much again for the amazing questions.
Thank you so much. Yeah, have a
Thank you,
beautiful day
Maria.
ahead. Thank you.
Okay.
Bye
Have a
bye.
good rest of the day
Bye.
or evening. Bye.